interesting talk. I'd like to invite our next speaker, Professor Valery Krizhanovsky from the Weizmann Institute of Science, to talk about the molecular mechanisms of senescence on the crossroads of cancer and aging.
This is the one, right? Okay, Valery, this is the one. Yeah. Okay, it's almost the end of the day, and I'd like to thank the organizers for inviting me to this, I think, first live conference for me after a long break. That's very exciting. And our lab is interested in cellular senescence, and as we have seen from the beautiful picture of hallmarks of aging that Ellie have shown us about an hour ago, senescence is one of the hallmarks of aging, or one of the cellular hallmarks of aging. But today I'd like to focus on a coordination between cancer and aging and the role of senescence in this. And I would like to start with this slide showing that cancer, or many types of cancer, is an age-related disease, and they rise with age, as for example, neurodegenerative diseases we talked about a lot. And we looked at one specific example of cancer, is pancreatic ductal adenocarcinoma, and you can see on the left side how incidence increases dramatically with age, and how after a long time of attempts, the treatment options are not very good, and it would be beneficial to understand what happens at the very beginning of the process of development of this cancer, and it might be we would be able to better diagnose it earlier, or by these means even prevent it. So why I think it would be possible, and this is because senescence is one of the things that drives different age-related diseases, including cancer, as was shown with this study, which is now already 5 years old, by Jan van Deursen and colleagues, where they did a pharmacogenetic trick and eliminated senescent cells in the whole mouse, and mice that don't have senescent cells since mid-age basically live significantly longer, their health span is extended, and they have and they have delayed appearance of many diseases, including cancer, because mice, just like humans, so much of their mortality is explained by cancer. Not the same types, but still. So basically, in this mouse model, elimination of senescent cells delayed cancer onset. But why would it happen? So what we know about senescence that would suggest to the mechanism of this effect. So just to have a quick recap, senescence is a terminal cell cycle arrest that can be induced by different types of stress listed here, and then activation of p53 and Rb mainly pathways. And in the short term, senescence has a positive protective role in induction of tumor suppression, in limiting tissue damage, and in guiding some structures during embryonic development. But then once senescent cells accumulate with age, and we know that they accumulate with age in many different organisms, including mice, humans, and so on, by producing what's called SAS, which is senescence-associated secretory phenotype, which is a plethora of cytokines and matrix remodeling enzymes. They can promote tumorigenesis, mainly in their microenvironment, and promote tissue aging and other age-related diseases. for a while, we have studied and we continue to study how immune system actually counteracts this accumulation of senescent cells and uncouples positive short-term responses from the deleterious long-term responses. And quite a few years ago already, we thought how we can, mimic this action pharmacologically or genetically, and to find ways for directed elimination of senescent cells. And we discovered a few pathways that can do it, and one of this is BCL2 family of proteins. We identified that BCL2 family of proteins are upregulated in senescent cells, and we can kill senescent cells using the inhibitor ABT737 that inhibits this BCL2 family of proteins. And this is one slide from this study, just showing that we can specifically eliminate senescent cells with this drug, and it doesn't matter what is the cause of senescence. It's damage-induced senescence, replicative senescence, or oncogene-induced senescence, which is more relevant to our study today. So with this tool in hand, we can ask now, what is the role of senescent cells in the process of tumorigenesis if we would be able to intervene early? And here is the time to look at this scheme where we see what happens in premalignant lesions, and this is classical, published already 15 years ago. We can see that if there is a genetic mutation in an oncogene, for example, RAS in one of the cells depicted in blue, then the cells would aberrantly proliferate, form a premalignant lesion, which is filled with senescent cells, but eventually one or more cells would escape this response and form tumor. Now, we can now intervene and eliminate senescent cells in this premalignant stage and ask what would happen to tumor development. And now back to the pancreatic cancer model, and we use two genetic models, one in our lab and one is a beautiful collaboration with Itai Ben Porat from Hadassah Medical School of Hebrew University and colleagues, and these are basically two genetic models of pancreatic cancer development that go through same route where in premalignant lesions called penins, senescent cells are present and we can identify them. For example, here using method we developed is using image stream that you can see on the right side, looking at which is cell type that we're looking at the cells are positive for senescence associated beta-galactosidase, that this is one of the markers of senescence, and we can see that if we're looking at the section, this is on the left, that the cells are also positive for drug targets, basically for BCL2 family of proteins, meaning we can actually target them with this drug. Now, are these cells the one that can promote tumor development in these lesions through their pro-inflammatory nature? So the answer is yes. So these cells express COX-2, which is one of the enzymes that you know is a target of many anti-inflammatory drugs, and it drives expression of many inflammatory cytokines. And this is in premalignant lesions in mouse and in human, and we can see co-staining of COX-2 with the marker of senescence. And here in one of our mouse models, we can see that these COX-2 positive cells accumulate in the premalignant lesions and in the relevant model, and they are not co-stained with the marker of proliferation, suggesting in a different way that they are that COX-2 is expressed in senescent cells. Now, can we actually eliminate these cells using the ABT737 drug? So now, if we treat the mice with ABT737, we can see increase in markers of apoptosis, here Clift caspase-3 in this premalignant lesion. We can see reduction in the presence of SA-beta-gal positive cells and reduction in presence of COX-2 positive cells measured by two different methods. So looking at this, we can eliminate senescent cells from premalignant lesions using ABT737. And here we also used model, like in vitro model, to see which cells we're eliminating. And this is a model of pancreatic ductal organoids. So these organoids apparently have some of the cells are senescent. We will see next slide. And now if we treat these organoids with ABT737 in presence of DAPI, we can see that the organoids that do have senescent cells, ones with expressing RAS, there are some cells are dying and they become DAPI positive, and you see a blue staining. Now, if we're looking at these organoids, so some of them express markers of senescence, like P21 here, and they also organoids derived from KRAS expressing mice have elevation of BCL2 family members and the markers of senescence like P21, P53, and SA-beta-gal. And if we treat these organoids with ABT737, we can see reduction in beta-gal positive cells. But we can also look at the organoids using single-cell RNA-seq and then look at the cells with the signature of proliferation or cells that are arrested. And these are vehicle-treated cells in the middle and ABT737-treated cells on the right, and you can see that most of the arrested cells are eliminated. So we can specifically eliminate senescent cells using ABT737 from premalignant lesions and also in culture. And now what would happen if we do it in a mouse model that we have seen in the beginning. So this is the treatment regimen is on the right top. We treat once in a month for 4 months, and then we look at the mice, at their pancreas, and we can see that the penins, the premalignant lesions, are significantly reduced, and the architecture of the pancreas is normal. And what's important, their SASP signature is reduced here by the cytokine array. But more interesting, what happens with the tumor development along the way. So here, again, the same treatment, once a month, it's actually twice, 2 consecutive days, and this kind of treatment once a month for 4 months. And this is at a time when there's still no tumors, only premalignant lesions. And then looking here at what's labeled as point 2, which is 4 months later on, when in the control mice, there are tumors already in about 70% of the mice, but in a mice that were treated at the premalignant stage, there's significant reduction in the amount of the tumors. About 25% of the mice only develop tumors, and there is if we measure the pancreas or ratio pancreas to the body weight, it's obviously reduced. But now, if we're looking at the sections of this pancreas, we can see that there is a reduction not only in the amount of the cancer, but also in the amount of different premalignant lesions, both penins and ADMs, which stands to SNR to ductal metaplasia. So basically, treatment that is performed at the premalignant stage in this genetic model where RAS is still active during all this period of time that we are looking at, is able to significantly reduce the development of cancer later on. So now to some let's say conclusions at this stage. So we have seen that senescent cells are present in the premalignant pancreas, then that they can be eliminated using the inhibitors of BCL2 family members, and that we can eliminate senescent cells only with treatment only at premalignant stage, and this treatment will block cancer development later on. Now, just to say again how we think it happens, so senescent cells are formed in these premalignant lesions as a protective response in response to RAS, what's called oncogene-induced senescence, and their initial function is to limit tumor development in response to activated oncogene, and they accumulate in this premalignant lesion, but they have pro-inflammatory signature that can promote tumorigenesis from the neighboring cells. Now, if we would extrapolate this model or kind of look at this back in terms of other age-related diseases, saying that this model is just another model of age-related disease, and would we be able to eliminate senescent cells once they're formed in order to prevent their further long-term negative consequences. And one of the ideas that one can think of, that normally senescent cells accumulate with age in the organism, immune system tries to keep them in check, and we are looking at this in many different ways, but both production of senescent cells can increase with age, and as we all know now with corona, we were all well aware the function of immune system goes down with age, so these two factors lead to further accumulation of senescent cells, which can contribute to a variety of age-related pathologies, and we can think about different ways to eliminate senescent cells or block their negative influence on their microenvironment. With this, I would like to thank people who did the work. This from my lab, it's Lior and Yossi, and for wonderful collaborators at Hadassah Medical School, and Itai Tirosh from Weizmann for the analysis of single-cell RNA-seq, and Fardin sources, and thank you very much for your attention.
Thank you for that fascinating talk. We actually have time for a question, if anybody has any questions to ask. Okay, well, thank you again. Oh, we do have a question, sorry, over here.
I just wonder, regarding the possibility that there is a strain-specific phenotype within mice, because we encounter strain-specific phenotypes in our studies from time to time. And I wonder if you tested in more than one strain of mice.
Yeah. So some of the, not all of the experiments, but some of the experiments were done on two different strains of mice, one in our lab at Weizmann and the other in Itai's lab at Hadassah, right? So, and most of the, but the, if you are asking about the end stage phenotype, so the second model is such that mice die before they reach to this stage because of pancreatic dysfunction. So it was only possible to test the premalignant stages in both models, and the tumor development was possible to test in only PDX KRAS model and not PTF1 alpha KRAS model.
Okay, thank you once again.